All right, let's dive straight into renal physiology. Welcome to this explainer. Today, we're setting up a premium, rock-solid academic foundation focused entirely on the incredible, high-pressure machinery of the human kidney. We're going to look closely at how this organ operates, not just as a simple biological filter, but as an absolute master regulator of your entire systemic homeostasis. Here is our roadmap for today, and it follows a really nice macro to micro progression. We'll kick things off with the core functions of the kidney, then break down the four sequential steps of renal function. After that, we'll compare plasma and urine to prove exactly how much alteration is actually happening. Then, we'll zoom way in on the microscopic functional anatomy of the nephron and wrap up by analyzing the kidney's absolutely massive blood supply.
Section 1. Core functions of the kidney, beyond just filtration.
It is super critical to recognize right from the jump that the kidney's role goes way, way beyond just making urine. Its impact is massively systemic. Looking at this list of responsibilities, first, yeah, of course, they excrete metabolic wastes and foreign chemicals. But second, they precisely regulate your body fluid osmolality and electrolytes, basically your entire water and salt balance. Third, they directly regulate arterial pressure. Fourth, they team up with the respiratory system to maintain your acid-base balance. Fifth, remember, the kidney is an endocrine organ too. It secretes, metabolizes, and excretes crucial hormones like erythropoietin for making red blood cells, 1,25-dihydroxycholecalciferol, which is your active vitamin D, and renin for blood pressure control. And finally, number 6, gluconeogenesis. I really want to pause on that last point for a second because it is a classic high-yield board exam trap. Gluconeogenesis is the synthesis of glucose from amino acids and other precursors. During prolonged fasting or starvation, your kidneys actually step up as a metabolic rescue organ. They literally rival the liver in their ability to synthesize brand new glucose just to keep your brain and your body fueled up. Definitely do not forget about that metabolic capability.
Ultimately, because of all these complex jobs, the final composition of your urine can vary wildly. And that's because many of your homeostatic regulatory mechanisms use the kidney to essentially buffer and protect your extracellular fluid. They alter the urine to keep the ECF perfectly stable. Once this highly modified fluid is finally formed, it takes the macroscopic path. It passes from the renal pelvis, flows down the ureters, hangs out in the bladder for a bit, and is finally expelled to the exterior via urination, which we also call micturition.
Moving on to section 2, 4 steps of renal function, the journey of the filtrate. To really understand how the kidney pulls off this whole extracellular fluid homeostasis thing, we have to look at 4 very precise sequential steps. Step 1, the glomeruli form an ultrafiltrate out of the blood plasma, which then flows right into the renal tubules. Step 2, as that fluid moves along, the tubules reabsorb solute and water. That means taking vital, good stuff back into the blood. Step 3, the tubules secrete solute and water. This is the opposite, moving specific substances from the blood directly into the tubular fluid to be trashed. And step 4, the final tubular fluid is excreted out of the body entirely. You absolutely must keep the distinction between reabsorption, saving things, and secretion, throwing extra things away, perfectly clear in your mind.
If we map those functional steps anatomically on this diagram, arrow 1 up at the top represents glomerular filtration, literally pushing plasma into the tubule. Arrow 2 shows tubular reabsorption, pulling fluid and solutes out of the tubule and bringing them safely back into the peritubular capillaries. Arrow 3 is tubular secretion, moving select solutes from those capillaries straight into the tubule. And lastly, arrow 4 represents the net result leaving the nephron, which is excretion.
Section 3, comparing plasma and urine, proof of alteration. Okay, so now that we've got the abstract concepts of filtration, reabsorption, and secretion down, we have to ask a pretty fundamental question. How drastically does the kidney actually alter this fluid? From its starting point as blood plasma to its final form as urine, just how powerful is this machinery? Before we look at the raw numbers, let's just quickly lock in those definitions one last time so there's zero confusion. Glomerular filtration filters the plasma. Easy enough. But as that filtrate passes down the tubules, its volume shrinks massively, and its composition is totally altered by tubular reabsorption, again removing water and solutes from the fluid to keep in the body, and tubular secretion, which is adding solutes into the fluid. Now check out this data. This is literally physiological magic at work. If we compare the concentration of substances in the plasma against the urine, let's focus on that U/P ratio, the urine to plasma ratio. Take glucose, for example. the plasma concentration is 100, but in normal urine, it's 0. That's a U/P ratio of exactly 0. Why? Complete 100% reabsorption. The kidney saves all of the fuel, no waste. On the flip side, look at creatinine. Plasma concentration is 1, but the urine concentration is 150. A U/P ratio of 150. Because creatinine is filtered but not reabsorbed, and since the tubules aggressively reabsorbs water along the way, that creatinine gets massively concentrated for elimination. It is a beautiful system, eliminating waste while perfectly conserving your vital metabolites.
Section 4. Functional Anatomy of the Nephron, the Microscopic Engine. To fully grasp how this drastic plasma alteration physically happens, we need to talk about scale. The macroscopic kidney is surprisingly small. It's about 10 centimeters long, 5.5 centimeters wide, and weighs only about 150 grams. But packed inside that tiny 150-gram organ are roughly 1 million individual nephrons. 1 million! And each one of those nephrons, which includes a renal tubule and its glomerulus, operates as its own independent, functional microscopic engine.
Visually tracing this path down from the macrostructure, you have the cortex on the outer rim, which is where the glomeruli sit, draining down into the medullary pyramids. If we zoom way into the cortex, we find the glomerulus itself. It's this intricate, microscopic tuft of capillaries that is perfectly designed for high-volume, continuous filtration. Understanding this glomerulus is absolutely key. Structurally, it's an invagination of a tuft of capillaries, lined by just a single layer of endothelial cells, resting on a specialized basement membrane. But here is a massive, highly tested board concept. Compared to pretty much any other capillary bed in your body, glomerular capillaries maintain an exceptionally high hydrostatic pressure. We're talking about 60 millimeters of mercury. That extreme pressure is the main physical driving force that literally shoves plasma out of the blood and into the tubule. Naturally, these highly pressurized, leaky capillaries don't just spray fluid into nowhere. They are completely encased in something called Bowman's capsule. Bowman's capsule is made of 2 layers, a visceral layer, which is the inner layer directly hugging the epithelial cells of the capillaries themselves, and a parietal layer, which acts as the outer casing. The space between those 2 layers catches all that newly formed ultrafiltrate, and that exact space physically continues straight down to become the lumen of the proximal convoluted tubule. Now, you might be wondering, how does the glomerulus manage to build up that crazy 60 millimeters of mercury of pressure in the first place? Well, blood enters the capillary tuft through the afferent arteriole. But take a look at the exit. It drains into the efferent arteriole, which has a slightly smaller diameter. By forcing high volumes of blood in and then slightly choking off the exit, it acts exactly like a dam. That's what builds up the critical hydrostatic pressure needed to force filtration. It really is a structural masterpiece. Section 5. Renal blood supply dynamics, feeding the filter. Obviously, sustaining 1 million of these high-pressure microscopic engines requires a vascular network that is just staggering. 1,200 milliliters per minute. Really think about that number for a second. Your kidneys receive 20 to 25%, a full 1/5 of your entire resting cardiac output. For a pair of organs that weigh just 150 grams each, that is the highest tissue-specific blood flow anywhere in the human body. All that massive volume of blood enters exclusively through the renal artery. From there, it branches out intricately, going from segmental to interlobar to arcuate, and finally to the interlobular arteries. This tree perfectly routes the blood up toward the outer rim of the kidney, precisely feeding each and every one of those individual afferent arterioles we just talked about. So where exactly does all that blood go once it's inside? This is another crucial physiological concept to lock in. 98% of total renal blood flow goes strictly to the renal cortex, while a measly 2% goes to the renal medulla. Why such an extreme unbalanced split? Because the cortex is where the glomeruli live. The cortex is doing all the heavy lifting of ultrafiltration, which demands an overwhelming supply of blood. Meanwhile, the medulla operates in a relatively low-flow, hyperosmotic environment, which is what allows it to concentrate the urine. So, as we wrap up this foundation, I'll leave you with this to chew on. With 1,200 milliliters of blood rushing in every single minute, relying entirely on a delicate, perfectly tuned 60 millimeters of mercury to filter and balance your entire physiological system, what catastrophic cascade happens to your body if this microscopic high-pressure filtration system fails? We've built the normal physiological foundation today, but understanding that failure is going to be the key to our future pathophysiology explainers. Thank you so much for watching. Definitely keep reviewing these mechanisms, and I'll catch you in the next one.
Welcome back, everyone. In this explainer, we're going to completely master the second half of renal physiology. We're moving step by step from the microscopic anatomy of the renal tubule all the way to the vital regulatory power of the juxtaglomerular apparatus. If you're a second-year medical student, this is the exact high-yield content you need to deeply understand how the kidney doesn't just filter your blood, but actively monitors and regulates your entire systemic blood pressure. So let's jump right in.
Here's our roadmap for navigating the complex machinery of the kidney today. We're covering the types of nephrons, the renal tubule pathway, the collecting system, JGA anatomy, JGA control mechanisms, and then we'll wrap up with a high-yield exam review.
Section 1. Types of nephrons. Think of these as the actual factories of the kidney.
Now, we begin our journey by looking at two distinct structures that dictate exactly how our bodies filter and concentrate fluids. The essential takeaway here is that we have two main categories of these factories: cortical nephrons and juxtamedullary nephrons. The really abundant cortical nephrons make up about 70 to 80% of the total nephrons. Their glomeruli are located right near the surface of the kidney, and their main gig is the bulk absorption of sodium. On the flip side, the juxtamedullary nephrons make up that remaining 20 to 30%. Their glomeruli sit much deeper, right at the junction of the cortex and the medulla. And as we're going to see, their highly specialized structure is exactly what allows them to concentrate your urine.
It's absolutely fascinating when you look at the physical difference in their blood supply. Notice how the juxtamedullary nephron has these incredibly long efferent arterioles extending from the glomeruli all the way down into the outer medulla. These vessels then divide into a specialized network of peritubular capillaries called the vasa recta. These vasa recta extend deep into the medulla, lying right side by side with those really long loops of Henle, before finally looping back up to empty into the cortical veins. This exact anatomical setup, it's the absolute essential physical requirement for the formation of concentrated urine.
Moving right along to Section 2, the renal tubule pathway. This is essentially our microscopic assembly line.
Literally every single drop of filtrate has to pass through this rigorous structural pathway, which is composed mainly of three parts. First, it hits the proximal convoluted tubule, then it dives into the loop of Henle, which is structurally divided into a thin segment, that's the descending limb, and the lower end of the ascending limb, where the walls are super thin, and a thick segment, which makes up the upper part of the ascending limb. Finally, the fluid moves into the distal convoluted tubule. The distinct cellular thickness of each of these segments directly dictates its highly specific physiological role in reabsorption and secretion.
Tracing this complete anatomical journey gives us a perfect visual of exactly where the thin descending limb structurally transitions to the thick ascending limb before hitting the distal tubule. You can actually just follow the filtrate leaving the renal corpuscle, weaving through the convoluted proximal tubule, dropping way down the loop, coming back up through that thicker ascending portion, and finally winding through the distal tubule.
Okay, Section 3, the collecting system, which handles the final transport.
After the primary nephron segments do all their intensive processing, the remaining fluid enters a highly organized drainage network. This is where the final modifications and transport of urine happen. Now, following this exact sequence is incredibly high yield for your exams. The distal convoluted tubules first open into initial arched collecting ducts called cortical collecting ducts, located in the renal cortex. Here's a crucial detail. 7 to 10 of these cortical ducts actually merge to form a single straight collecting duct that passes into the medulla, becoming a medullary collecting duct. Down in the inner zone of the medulla, these merge even further to form papillary ducts, also known as the ducts of Bellini. These empty into the papilla of the minor calyces. 3 or 4 minor calyces unite to form one major calyx, which then opens into the expanded renal pelvis and finally continues out of the kidney as the ureter.
And you know, all that structural depth we just talked about, it translates directly to physiological concentration gradients. As the collecting duct plunges through the inner zone of the medulla, the surrounding tissue osmolarity increases drastically. We're talking from 300 milliosmoles per liter in the cortex all the way up to 1200 deep in the medulla. When water channels are open, water leaves the collecting duct by osmosis right into this hyperosmotic interstitium, which powerfully concentrates the urine before it even leaves the body.
All right, Section 4, Juxtaglomerular Apparatus Anatomy, the master sensor. We have now arrived at the real climax of this explainer, the microscopic control center that connects tubular flow directly back to systemic cardiovascular regulation. So let's dive into this incredibly specific cellular modification. As the initial portion of the distal tubule loops back around, it nestles perfectly right between the afferent and efferent arterioles of its own parent glomerulus. At this exact physical contact point, the tubular epithelial cells are histologically modified to form what we call the macula densa. This strategic anatomical placement is exactly what allows the macula densa to sort of taste and monitor the sodium concentration of the filtrate right before it leaves the main processing line.
Now, you absolutely gotta know the three distinct cellular players that make up the entire juxtaglomerular apparatus complex. First, we have the macula densa we just talked about. Second, we have the actual juxtaglomerular cells, or JG cells, which are specialized cells located within the walls of the afferent arteriole that actually secrete renin. And third, we have the Lassie cells, also known as extraglomerular mesangial cells. These are flat, elongated cells located near the macula densa, and honestly, while they are a part of the complex, their exact function still remains largely unclear in medical physiology.
When you look at the physical reality of this system, you can clearly see those modified columnar cells of the macula densa sitting inside the wall of the distal tubule. Notice how it's nestling right up against the smooth muscle-derived juxtaglomerular cells of the afferent arteriole. Because they physically touch, they form this direct physical feedback loop. It's brilliant, really. It allows chemical signals from the tubule to instantly alter the behavior of the blood vessel.
Which brings us to Section 5. JGA Physiological Control Mechanisms.
Let's see how this structural intimacy builds into an active, dynamic response system when your body's chemistry or blood pressure changes. Let's break down exactly how the macula densa acts as a chemical sensor. When it detects an elevated sodium concentration in the tubular fluid passing by, it interprets this as the filtrate moving way too fast for proper absorption. In response, these cells trigger a direct contraction of the adjacent afferent arteriole. By constricting this incoming vessel, blood flow into the glomerulus drops, which effectively lowers the glomerular filtration rate, or GFR. It literally pinches off the incoming blood supply to slow down the whole filtration factory just so the tubules can catch up.
On the flip side, the JG cells act like microscopic pressure sensors. Because they're located right in the walls of the afferent arteriole, they stretch and relax with your blood pressure. If the JG cells feel that the incoming blood pressure is too low, they actively secrete the enzyme renin directly into the bloodstream. This secretion of renin kicks off the massive renin-angiotensin-aldosterone system, or RAS, which acts throughout your entire body to clamp down on blood vessels and retain salt and water, ultimately rescuing your systemic blood pressure.
Finally, Section 6, High-Yield Exam Review.
Let's lock in these details with a rapid-fire revision of the most heavily tested concepts from this half of the explainer. Definitely take a screenshot of these points. They represent the absolute core physiological relationships you're going to be tested on. Remember your ratios. Cortical nephrons dominated 70 to 80%, but those 20 to 30% of juxtamedullary nephrons with their exclusive vasa recta are entirely responsible for urine concentration. Remember that 7 to 10 cortical collecting ducts merge into one straight duct. And finally, lock down your sensors. The macula densa detects sodium to control GFR locally, while JG cells detect pressure to control systemic blood pressure via renin.
I'll leave you with this final highly practical exam tip to keep these regulatory systems perfectly straight in your mind. Just ask yourself on test day, which sensor controls which pathway? The macula densa is your chemist, checking the sodium levels. The JG cells are your pressure gauges, feeling the stretch of the blood vessels. Master this distinct anatomy, and you will understand exactly how the kidney performs these physiological miracles. But consider this before our next explainer. What happens to your entire cardiovascular system when this delicate microscopic pressure sensor malfunctions during chronic hypertension? Keep studying hard, and we'll explore that next time.
Welcome to this high-yield explainer on renal physiology. Today, we're tracing the microscopic and mathematical journey of the renal filtrate. Since you're already second-year med students, I know you've got the foundational anatomy down pat. So we're jumping straight into the deep end of the physiological mechanisms. We're going to map out the precise functional pathways, starting all the way from vascular regulation right down to the math of clearance. Let's get into it. Here's our roadmap for today. We'll cover the renal sympathetic nerve supply, the 4 core nephron processes, glomerular filtration and its barrier, GFR and filtration fraction, the exact pressures driving that filtration, and finally, the principles of renal clearance. Starting off with Part 1, renal sympathetic nerve supply. Take a look at the targeted precision of the kidney's autonomic wiring here. The kidney receives a really rich sympathetic noradrenal innervation. When the adrenal gland and sympathetic nerves release norepinephrine, it hits 3 specific receptor types:
each with its own crucial job. First up, alpha-1 receptors on the vascular smooth muscle. These cause vasoconstriction, which controls our renal blood flow. Then we've got beta-1 receptors sitting on the juxtaglomerular, or JG cells, and these are the ones that trigger renin secretion. Finally, alpha and beta receptors right there on the renal tubules kickstart sodium and water reabsorption. Oh, and here's a massive takeaway for your exams. There is basically no significant parasympathetic innervation in the kidneys. The control here is overwhelmingly sympathetic.
Moving right along to Section 2, the 4 core nephron processes.
All right, so every single substance the kidney handles goes through some combination of 4 core processes. Step 1 is filtration, right? That's fluid moving from the glomerular capillaries into Bowman's capsule. Step 2 is reabsorption. Think of this as a massive reclamation project where all the good stuff, the essential substances, moves from the tubular fluid back into the peritubular capillaries. Step 3 is secretion. This is an active transport process where the body forcefully moves additional waste from those peritubular capillaries straight into the tubule. And step 4, that's urinary excretion, the final removal of whatever fluid and solutes are left over.
Now, how do those core pathways actually combine in real life? Well, there are 4 main physiological scenarios. Substance A over there, that is filtration only. Whatever goes into the tubule leaves in the urine, which makes it a perfect marker for measuring flow. Then you have substance B. It gets filtered, but then undergoes partial reabsorption. Basically, the blood takes some back, but a portion still gets excreted. Substance C is something the body really wants to hold on to, like glucose or amino acids. It gets filtered, but then it's completely reabsorbed. Under normal conditions, you won't see a drop of it in the urine. Finally, substance D. It's filtered, sure, but the body really wants it gone, so it actively secretes even more of it from the blood into the tubule to clear it out as fast as possible.
Zooming strictly into Bowman's capsule, we hit Section 3, glomerular filtration and barrier.
You absolutely need to know this foundational principle. Glomerular filtrate is practically identical to blood plasma, with one huge exception. It doesn't have any significant amount of proteins. So how does the body pull off that neat trick? It is because the glomerular membrane is incredibly selective. And that selectivity relies heavily on just 2 physical properties of the molecules trying to cross, their molecular size and their electrical charge.
For plasma to make it from the capillary lumen into Bowman's space, it has to run the gauntlet through 3 distinct structural layers. Think of it like a highly sophisticated microscopic sieve. First, it hits the fenestrated endothelium of the capillary, then the basement membrane, and lastly, the slit pores of the visceral epithelial cells. Let's visualize that architecture. Down at the bottom, see those fenestrations? They are literally physical windows inside the endothelial cells. Right above that is the basement membrane, an acellular matrix that acts as a huge electrical charge barrier, essentially repelling negatively charged proteins. Finally, wrapping around the capillaries, you have the podocytes. These visceral epithelial cells have these incredibly intricate foot processes that interlace with each other to form really narrow slit pores. Together, the whole shebang forms our definitive, highly selective barrier.
All right, Section 4, GFR and filtration fraction. Let's look at the numbers.
You've probably heard of the glomerular filtration rate, or GFR. It is simply the total volume of fluid filtered through the glomerulus into Bowman's capsule every single minute across all nephrons in both of your kidneys. In a healthy adult, that engine is generating a massive 125 milliliters per minute. If you scale that up, that is roughly 180 liters per day. I mean, just think about that for a second. Your kidneys are filtering the volume of a large bathtub every single day. Crazy, right?
But wait, we have to put that number into perspective with our total blood flow. Normal renal plasma flow is about 650 milliliters per minute. So if we are filtering out 125 milliliters from that 650, we get what is called the filtration fraction. It's the actual percentage of plasma that gets turned into glomerular filtrate. Do the math, and it averages out to about 1/5, or exactly 19%.
Let's trace that circulatory pathway and round the numbers a bit to make it super intuitive. Out of the 100% of plasma entering the afferent arteriole, 80% just waves goodbye and continues straight into the efferent arteriole. The other 20% gets filtered into Bowman's capsule. But hey, remember that massive 180-liter bathtub we talked about? If we actually excreted all of that, we'd literally dehydrate in a matter of hours. So out of that 20% that gets filtered, more than 99% is meticulously reabsorbed by the rest of the nephron. End result? Less than 1% of that total volume actually leaves the body as urine.
Moving to Section 5, pressures driving glomerular filtration.
Okay, so what actually pushes and pulls that fluid across the barrier? It comes down to 2 major factors. The first is the capillary filtration coefficient, basically how permeable the capillaries are and their total surface area. The second is the driving force itself.
This is a really delicate tug-of-war of hydrostatic and colloid osmotic pressures located in both the capillary and Bowman's capsule. To be specific, you've got glomerular capillary hydrostatic pressure forcefully pushing fluid out, Bowman's capsule hydrostatic pressure pushing back, glomerular capillary colloid osmotic pressure pulling fluid back in, and finally, Bowman's colloid osmotic pressure. Now, that last one is normally 0 because, like we said earlier, large proteins aren't crossing that barrier anyway. Let's do the math on those opposing forces. We start with our big favoring force, the glomerular hydrostatic pressure, sitting at 60 millimeters of mercury. From that, we have to subtract the forces fighting against it. That's 18 millimeters of mercury from Bowman's capsule hydrostatic pressure and 32 millimeters of mercury from the glomerular capillary colloid osmotic pressure. So 60 minus 18 minus 32 leaves us with exactly 10 millimeters of mercury. That is our net filtration pressure, just 10 millimeters. It's wild to think that this tiny 10-millimeter gradient is the sole engine driving that entire 180-liter daily filtration.
Which brings us to our final part today, Section 6, Principles of Renal Clearance.
Conceptually, what is renal clearance? It's defined as the volume of blood plasma from which a specific waste substance is completely and perfectly removed in exactly 1 minute. It is absolutely crucial to grasp this. Clearance is a measurement of the plasma volume that gets cleared. It is not a measurement of the physical amount of the substance itself. Keep that straight in your head. So, linking this back to those 4 macroscopic processes we discussed earlier, this clearance value is really just the net mathematical result of 3 things. It's the initial glomerular filtration of the waste, plus whatever amount gets actively added via tubular secretion, minus whatever amount gets reclaimed back into the blood by tubular reabsorption.
Now, if we want to accurately measure GFR clinically, we need an ideal clearance substance. On the words, you'll see things like urea, creatinine, and para-aminohippuric acid used. But for a substance to be a perfect marker, like inulin, it has to meet some really strict criteria. It must be freely filtered at the glomerulus, completely ignored by the tubules, meaning no reabsorption and no secretion. It can't be metabolized or stored in the kidney. It has to be entirely non-toxic, and, perhaps most importantly, its presence absolutely cannot alter the GFR itself.
And that brilliantly brings us to the fundamental equation you are going to use over and over again on the wards. Serum C equals U times V divided by P. Let's break it down. Clearance, or C, equals the concentration of the substance in the urine, that's our U, measured in milligrams per milliliter, multiplied by the urine output volume, which is V, in milliliters per minute. So you take that total excreted amount and divide the whole thing by its concentration in the plasma, which is P. This elegant little formula lets us precisely quantify a patient's kidney function using nothing more than a blood draw and a timed urine sample. Pretty awesome, right? Well, we've covered a lot of ground today. We traced the journey from the sympathetic nerve wiring through the microscopic sieve of the filtration barrier, calculated that 19% filtration fraction, and wrapped up with the clinical math of clearance. But I want to leave you with a really profound physiological puzzle. If our massive 180-liter daily filtration relies entirely on that super delicate 10 millimeter of mercury pressure gradient, how in the world do the kidneys autonomously defend that tiny gradient to prevent sudden, catastrophic renal failure whenever our systemic blood pressure drops? Ponder that autoregulation for a bit, and I'll see you in the next explainer. Welcome, second-year med students, to this explainer on renal physiology. Today, we're going to master the delicate balancing act of renal blood flow, or RBF, and glomerular filtration rate, GFR. We're looking at how the kidney makes sure you don't literally pee your entire blood volume away when your blood pressure fluctuates. It's an absolutely brilliant piece of biological engineering, and honestly, by the end of this, you'll understand exactly how it all ticks. Here's our roadmap for this explainer. First, the stakes. Then, emergency override. The anatomy of control. Tubuloglomerular feedback mechanisms, metabolic influences, and finally, a summary and exam tips. Okay, part 1. The stakes. Why autoregulation matters. To really get why these mechanisms exist, we first have to look at the terrifying mathematical reality of what would happen without them. Notice the sheer scale of the disaster represented by this 30-fold number. Normally, your GFR is about 180 liters per day, and their tubules reabsorb 178.5 of those liters. That leaves a perfectly normal, manageable 1.5 liters of urine to excrete. But if you didn't have autoregulation,
Well, a minor blood pressure jump from 100 to 125 millimeters of mercury would push your GFR up to 225 liters a day. If tubular reabsorption just stayed constant, your urine output would skyrocket to 46.5 liters a day. That is a 30-fold increase. You'd deplete your entire blood volume in a matter of hours. So yeah, the kidney desperately needs safeguards. To prevent us from rapidly losing our blood volume like that, the kidney relies on 3 distinct negative feedback mechanisms, or pillars of control. We've got the neural factor, the hormonal factor, and intrinsic autoregulation. Let's unpack exactly how they share the workload.
Moving right along to Part 2, Emergency Override, Neural and Hormonal Factors.
Let's start with the emergency overrides. These are the neural and hormonal factors that kick in during acute, life-threatening crises. These aren't your day-to-day managers. These are the absolute alarm bells. First up is the neural factor. So all the blood vessels of your kidneys are richly innervated by sympathetic nerve fibers. During a severe acute disturbance, like, say, a severe hemorrhage, your sympathetic nervous system aggressively activates. It heavily constricts the renal arterioles. This immediately decreases your renal blood flow and your GFR. The kidney is essentially sacrificing its own filtration rate to decrease urine output and keep your overall blood volume within normal survivable limits. Hormonally, we have 2 teams here, the constrictors and the dilators. Norepinephrine, epinephrine, and endothelin constrict blood vessels and decrease GFR. In extreme conditions like that severe hemorrhage we just talked about, blood levels of norepinephrine and epinephrine perfectly parallel your sympathetic nervous activity. And hey, keep a really close eye on endothelin. Its levels increase pathologically in diseases like toxemia of pregnancy and acute renal failure, causing severe renal vasoconstriction. On the flip side, endothelia-derived nitric oxide decreases renal vascular resistance to increase GFR, alongside prostaglandins and bradykinin, which also tend to increase GFR. Now, the crucial point is knowing how to distinguish between these constrictors. And here is a classic exam trap that catches so many students: angiotensin II. Angiotensin II is a constrictor, yes, but unlike the others, it specifically targets the efferent arteriole, the one leaving the glomerulus. By pinching the exit pipe, it creates a back pressure inside the glomerular capillaries. This brilliantly illustrates how constricting a specific vessel actually leads to an increase in glomerular filtration rate. Absolutely do not forget that distinction.
All right, Part 3, the anatomy of control, autoregulation.
Now, let's transition from those systemic emergencies to the everyday heroes. These are the intrinsic autoregulatory mechanisms that keep your GFR relatively constant day to day, despite marked changes in your arterial blood pressure. Before we dive into the exact physiology, map this beautiful anatomy in your mind. This is the juxtaglomerular apparatus. Notice the macula densa. These are specialized epithelial cells sitting right in the wall of the distal tubule. Look at how perfectly positioned they are. They physically touch the angle between the afferent arteriole and the efferent arteriole. Nestled right there in the afferent arteriole are the smooth muscle juxtaglomerular cells. This anatomical proximity is definitely no accident. It's the physical hardware that makes autoregulation possible. Which brings us to Part 4, tubulo-glomerular feedback mechanisms. This precise anatomical setup makes the tubulo-glomerular mechanism possible. This mechanism provides the exact degree of GFR and RBF required, and it actually operates through 2 distinct, simultaneous feedback pathways. The first pathway is the afferent arteriolar vasodilator feedback mechanism. Imagine a patient has a low GFR due to dehydration. This causes a very sluggish, low flow rate of tubular fluid. Because the fluid is moving so slowly through the ascending limb of the loop of Henle, the cells have extra time to overabsorb sodium and chloride ions. By the time that fluid reaches the distal tubule, the ion concentration is abnormally low. The macula densa senses this low sodium and chloride. It immediately sends a signal to dilate the afferent arteriole. Opening that entrance pipe increases blood flow into the glomerulus, causing an increase in glomerular pressure and bringing the GFR right back up toward the required level. Simultaneously, we have the efferent arteriolar vasoconstrictor mechanism. That exact same signal, the low sodium and chloride ions at the macula densa, also stimulates the juxtaglomerular cells. These cells release an enzyme called renin. Renin acts on angiotensinogen in the blood, eventually causing the formation of angiotensin II.
And what did we say about angiotensin II earlier? It powerfully constricts the efferent arterioles way more than the afferent ones. This pinches the exit, causing an increase in the pressure in the glomerulus, driving the GFR back to normal.
Let's see how this builds into one complete picture. This beautiful flowchart brings it all together, visually proving how both mechanisms work in perfect harmony. When arterial pressure drops, GFR drops. The macula densa senses the resulting low sodium chloride. It then triggers both the afferent dilation to let more blood in and the renin-angiotensin efferent constriction to keep the blood pressure high inside the glomerulus. The ultimate goal of this renin-angiotensin activation is to perfectly conserve your water and salt while still maintaining enough filtration pressure to allow toxic waste products to be excreted. Beyond the tubuloglomerular feedback from the macula densa, we also have a second autoregulatory mechanism, the myogenic mechanism. This one is beautifully simple. It's just an intrinsic feature of vascular smooth muscle. When arterial blood pressure spikes, the high wall tension physically stretches the smooth muscles in the blood vessels. The smooth muscle simply resists that stretching by contracting. This prevents overdistension of the blood vessels, increases vascular resistance in the afferent arterioles, and physically blocks excessive increases in renal blood flow and GFR.
Part 5. Clinical Context. Metabolic Influences. Finally, let's ground this textbook physiology in real-world dietary and clinical contexts. What actually happens when we introduce high levels of protein or glucose into this highly calibrated system? Well, when you eat a high-protein meal, your GFR and RBF actually increase by 20 to 30% within just 1 or 2 hours. Why? A high-protein meal increases amino acids in your blood. In the proximal tubule, these amino acids are reabsorbed together with sodium. Because the proximal tubule is greedily pulling all this extra sodium out of the fluid to co-transport the amino acids, it ironically decreases the amount of sodium that gets delivered downstream to the macula densa. As we just learned, the macula densa panics when it sees low sodium. It triggers the tubuloglomerular feedback mechanism, incorrectly thinking GFR is low, and artificially drives the GFR up. And this exact same physiological hijack occurs in uncontrolled diabetes mellitus. Instead of amino acids, you have massive increases in blood glucose. The proximal tubule furiously reabsorbs this glucose, dragging massive amounts of sodium right along with it. Once again, the macula densa is starved of sodium, the tubuloglomerular feedback is triggered, and you see marked pathological increases in renal blood flow and GFR. This is a highly crucial concept for understanding diabetic nephropathy.
Wrapping up with Part 6, Summary and Exam Tips.
Okay, let's lock this knowledge in. We've covered the systemic overrides and the intricate local mechanisms. Remember these 5 absolute truths for your exams. First, without autoregulation, a small BP bump causes lethal fluid loss. Second, sympathetic tone and endothelium, think acute failure and toxemia, strongly drop GFR. Third, never forget the board trap. Angiotensin II constricts the efferent arteriole to raise GFR. Fourth, the macula densa is the master sensor. Low sodium chloride triggers afferent dilation and JG cell renin release. And fifth, high protein or high glucose paradoxically increases GFR by stealing sodium in the proximal tubule. Before you head back to the textbooks, consider this scenario. If a patient is bleeding heavily, triggering massive sympathetic vasoconstriction to save their life, but they also have severely uncontrolled diabetes, desperately trying to spike their GFR through macular densa feedback, which physiological force wins the tug of war over their glomerulus? Think about what the body prioritizes for immediate survival. It's a fascinating intersection of pathology and physiology. Keep studying hard, trust the structure of these mechanisms, and I'll catch you in the next explainer. Hey everyone, and welcome to this explainer. Today, we're literally following the filtrate on its exact journey out of the glomerulus, mapping every crucial step of renal reabsorption. If you are a second-year medical student looking to build a rock-solid, deep physiological understanding of how the nephron handles sodium and water, you are in exactly the right place. We are going to break down these complex cellular transport mechanisms step by step, so let's get right into it. We've got a very clear roadmap for today to make sure you master these high-yield concepts. We'll start with the macro basics, dive deep into the proximal tubule and its cellular transport, decode glomerulotubular balance, and finally unpack the unique fluid dynamics happening in Henle's loop.
Let's start with a macro view, the absolute basics of renal reabsorption. Just look at the sheer scale of work your kidneys are doing right now. An unbelievable 99.4% of the filtered sodium gets reabsorbed. Think about that workload. The proximal convoluted tubule alone is doing the heavy lifting, grabbing about 67% of it right out of the gate. Then, as we move down the line, the thick ascending limb handles 25%, the distal convoluted tubule takes 5%, and the collecting duct finishes up with 3%. That leaves less than 1% to actually be excreted in the urine. This breakdown really sets the stage for our detailed journey.
So let's zoom in on that heavy lifter, the proximal convoluted tubule. The absolutely crucial thing to remember about this segment is that reabsorption here is essentially isotonic. What does that actually mean for us? It means water directly follows the solutes. As massive amounts of sodium, chloride, and other vital solutes are actively pumped out of the tubular lumen and into the interstitial space, water passively tags right along at the exact same proportional rate. Because they move hand in hand, the osmolality of the tubular fluid stays identical to the plasma. It doesn't concentrate the urine, it doesn't dilute it, it just dramatically shrinks the total volume.
Now, to support this insanely rapid active transport, these epithelial cells are structurally geared up for the job. If you were to look at these under an electron microscope, you'd see they are absolutely packed with mitochondria. They have to be to generate the immense amount of ATP required to keep those pumps running nonstop. Plus, the luminal surface isn't just flat. It has an incredibly dense brush border made of microvilli. This exponentially increases the surface area so the cell can catch and pull in all those filtered solutes. It's a perfect example of structure dictating function.
All right, let's get into the nitty-gritty of how this actually works at the cellular level.
Check out what's happening on the basolateral side. That's the side facing the peritubular capillary. You've got this active sodium-potassium ATPase pump. It's actively burning energy to constantly throw sodium out of the cell. This is genius because it keeps intracellular sodium levels extremely low, creating a powerful concentration gradient. Then, on the luminal side, the cell basically exploits that gradient. Sodium naturally wants to rush downhill into the cell, and carrier proteins use that kinetic energy to drag glucose, amino acids, phosphate, and lactate inside, totally against their own gradients. This is your classic secondary active transport, and it absolutely dominates the first half of the proximal tubule.
Moving into the second half of the proximal tubule, the game changes just a bit. We see a huge reliance on a slightly different secondary active transport mechanism, one that depends heavily on the enzyme carbonic anhydrase. This ties directly into your acid-base balance. Inside the tubular cell, carbon dioxide reacts with water to form carbonic acid, catalyzed by that carbonic anhydrase. That acid then quickly splits into a hydrogen ion and a bicarbonate ion. Now the cell has a problem. It needs to ditch that hydrogen ion while saving the sodium. So it uses the energy from sodium moving downhill into the cell to force that hydrogen ion uphill and push it out into the lumen.
You can actually see this in action with the sodium-hydrogen exchanger on the luminal membrane. The downhill rush of sodium powers the uphill secretion of hydrogen. Once that hydrogen is out in the lumen, it hooks up with filtered bicarbonate ion to form carbonic acid. Then, a luminal version of carbonic anhydrase breaks it right back down into water and CO2. That CO2 just freely diffuses back into the cell and the whole cycle starts over. The end result here is brilliant. For every hydrogen secreted, we rescue one sodium ion. And, crucially, one bicarbonate ion gets reabsorbed back into the blood, which you know is absolutely vital for maintaining systemic pH. So by the time we reach the end of this tubule, about 60 to 70% of the filtered solutes in water have been moved into the interstitial fluid. This huge buildup of sodium, chloride, and bicarbonate drastically spikes the osmotic pressure in the ISF. That osmotic force is so strong, it literally pulls water out of the tubule. Once it's in the interstitial space, ultrafiltration takes the wheel. The high collate osmotic pressure and the low hydrostatic pressure inside the peritubular capillaries act like a giant vacuum, sweeping all that water and solute right back into the systemic circulation. Which brings us to a really elegant and highly testable physiological rule: glomerulotubular balance. The proximal tubule has this magical ability to maintain a proportional balance no matter what happens to your glomerular filtration rate. It stubbornly reabsorbs a constant 65% of the sodium and fluid. Let's look at the math. If your GFR is 100 milliliters per minute, the tubule reabsorbs 65. But let's say systemic changes cause your GFR to suddenly spike to 200. The proximal tubule doesn't just get overwhelmed, it scales its absolute reabsorption right up to 130 milliliters per minute. The absolute volume changes, but that percentage stays locked at roughly 65%.
This is a brilliant evolutionary design to prevent the distal segments of your nephron from getting completely flooded if your GFR suddenly shoots up. Now, as that remaining filtrate leaves the cortex, we plunge deep into the medulla, straight into Henle's loop, where the rules of fluid dynamics completely change. What you really need to notice here are the totally opposite permeabilities of the descending and ascending limbs. In the thin descending loop, you've got water freely leaving the tubule. But look at the thick ascending loop. The water stays put, and instead, you see sodium, chloride, and potassium being aggressively pumped out. This stark contrast in structural permeability is basically the entire foundation for renal physiology in the medulla. We've got a brilliant setup here. The descending limb is highly permeable to water, meaning it leaks water into the hypertonic medullary interstitium. Because it's losing water but keeping its solutes, the fluid inside gets highly concentrated, or hypertonic, right as it hits the hairpin turn. But as it turns that corner and heads up the thick ascending limb, the walls suddenly become strictly impermeable to water and urea. Instead of water leaving, the limb aggressively pumps solutes out. By stripping away solutes but locking the water inside, the tubular fluid actually becomes hypotonic relative to plasma. That's exactly why the thick ascending limb earned its clinical name, the diluting segment. Let's zoom in a bit closer on exactly how this diluting segment pulls this off. You've got to pay close attention to this specific luminal carrier. This is the famous sodium-potassium 2-chloride cotransporter. It literally grabs 1 sodium, 1 potassium, and 2 chloride ions and rips them all into the cell at the exact same time. Why is this so crucial for your exams? Because this specific carrier is exactly what loop diuretics like furosemide block. If you hit this transporter with furosemide, those ions get trapped in the lumen. The medulla can't build its hypertonic gradient, and as a result, the body is forced to excrete massive amounts of water. Now, just like in the proximal tubule, none of that fancy luminal transport works without the basolateral sodium-potassium ATPA pump on the other side. It's constantly bailing sodium out of the cell to keep the intracellular concentration ridiculously low. In fact, the thick ascending limb has a higher concentration of these energy-hungry pumps than literally any other part of the entire nephron. That's a huge clue telling us just how aggressive and vital solute reabsorption is right here. But we have a slight logistical problem here. There just isn't enough potassium in the filtrate to keep that triple cotransporter running continuously. So what happens? Once the potassium is pulled into the cell, it actually diffuses right back out into the lumen through passive channels. It recycles itself. Meanwhile, the 2 chloride ions head out the back door toward the blood. One diffuses passively into the interstitial fluid, and the other is cotransported alongside a potassium ion. This perfectly coordinated cellular dance ensures the solutes make it all the way to the peritubular capillaries. All right, let's wrap this up with a question to chew on. We've just seen massive isotonic bulk reabsorption in the proximal tubule, followed by the brilliant water-segregating mechanics of the loop of Henle that purposefully create a hypotonic fluid. So if the loop of Henle goes out of its way to build these extreme osmotic gradients, how exactly will the final segments of the nephron utilize those gradients to dictate your ultimate urine concentration? We've laid the foundation here. The stage is set perfectly for the final hormonal adjustments in the distal nephron, but we'll have to tackle exactly how aldosterone and ADH apply those finishing touches next time.
Welcome back to The Explainer. So think about this for a second. Your kidneys filter enough fluid every single day to fill an entire bathtub, right? But they only let about a liter actually escape as urine. It's kind of mind-blowing. Today, we're going to master exactly how the distal nephron achieves this magic. We'll focus on normal, healthy function and the major diuretic classes that are heavily tested on your medical boards. To do this, we're using what I like to call a baseline and intervention model. Basically, we're going to lock in the normal physiology of the nephron first. That's our baseline, and then we'll see exactly how specific pharmacological interventions disrupt that system. Let's get started.
Okay, let's take a quick look at our roadmap for today. We're covering the distal nephron, principal and intercalated cells, hormonal control, clinical diuretics, and then we'll wrap things up with a high-yield review.
All right, Section 1, the distal nephron, setting the anatomical stage.
So here's the thing you have to remember. You really can't treat the distal tubule as just one uniform tube. It simply doesn't work that way. The early segment, well, functionally, it's just an extension of the thick ascending limb. It continues this intense ion transport without letting any water through. But then, as we move into the late distal tubule and the cortical collecting duct, we see this major functional shift. This area houses two completely distinct, highly specialized cell types.
Moving right along to Section 2, Principal versus Intercalated Cells, the cellular workforce.
Now, what's really interesting here is the stark contrast in function between these cellular neighbors. On one hand, you've got your principal, or P cells. They're busy reabsorbing sodium and secreting potassium, and its entire operation is powered by the basolateral Na plus K plus ATPase pump, which creates that essential electrochemical gradient driving the transport. On the other hand, you have your intercalated, or I cells, which handle a completely different but absolutely crucial task: rigorous acid-base regulation.
Section 3, Hormonal Control Mechanisms, specifically aldosterone and ADH.
Okay, let's walk through this aldosterone cascade step by step, focusing first on those principal cells. Step 1. Aldosterone is secreted from the adrenal cortex. Simple enough. Step 2. Once it actually reaches the kidney, it gets right to work. It increases the number of active epithelial sodium channels. Those are your ENaC channels, right there on the luminal membrane of the P cells. And finally, step 3, it simultaneously cranks up the activity of the Na plus K plus ATP pump on the basolateral side. It's really like a master switch. This beautifully illustrates how just one single hormone perfectly sets up the entire cellular machinery required for powerful sodium reabsorption and potassium secretion.
But wait, aldosterone doesn't just stop at the P cells. If we look over at the intercalated cells, you'll see these critical subtypes for acid-base balance. Alpha intercalated cells secrete acid, hydrogen ions, using an ATP-driven proton pump known as the H plus ATPase. And guess what? Aldosterone acts on this specific pump too, increasing distal hydrogen secretion. Meanwhile, the beta intercalated cells do the exact opposite. They secrete base, or bicarbonate. Oh, and we absolutely can't forget antidiuretic hormone, or ADH. This puts the final touch on volume control. It drops aquaporin 2 channels into the luminal membrane, reclaiming about 5% of the filtered water right at the finish line.
Section 4, Diuretics and Clinical Interventions, Pharmacological Disruptions.
All right, let's talk drugs and see how this builds into our clinical interventions. Diuresis is fundamentally an increase in urine flow rate. How do we do it? By inhibiting luminal sodium reabsorption. That unabsorbed sodium basically acts like an osmotic magnet, literally trapping water in the tubule. Enter our first class of molecular saboteurs, loop diuretics, like furosemide. These guys specifically block the NKCC2 co-transporter in the thick ascending limb. Now, since this segment normally reabsorbs a massive 20 to 25% of our filtered sodium, blocking it causes an absolutely explosive increase in urine flow. Because it's so incredibly potent, furosemide's perfectly suited for something like rapidly clearing pulmonary edema.
Moving a bit further down the tubule, we hit thiazide diuretics, like hydrochlorothiazide. These target the NCC sodium chloride symporter in the early distal tubule. Now, thiazides are definitely less potent than loop diuretics. Why? Well, because a much lower proportion of the filtered sodium load is actually reabsorbed in the distal tubule compared to the loop of Henle. But even though they're less potent, they are fantastic first-line agents for treating hypertension, and they also help relieve edema and calciuria. There is a catch, though: potassium loss. They drop blood potassium in 2 indirect ways. First, by pushing more sodium downstream, and second, through secondary ROS activation because of volume depletion. That volume depletion stimulates aldosterone, which then hyperactivates the Na plus K plus ATPase downstream.
So to counter that dangerous potassium loss we just talked about, clinicians reach for potassium-sparing diuretics. These target the principal cells in the cortical collecting duct. The logic here is simple. By stopping sodium from entering the cell, less potassium and hydrogen ions are exchanged and lost to the urine. They do this in one of 2 ways. Mechanism 1 directly blocks the ENaC apical sodium channels. That's exactly how amiloride works. Mechanism 2 uses aldosterone antagonists, like spironolactone, to basically reduce the overall expression and activity of those sodium transport proteins. Now, because they act so far downstream where less than 5% of filtered sodium is being reabsorbed, they really only give you a very weak diuretic effect. But honestly, that's fine, because they are absolute masters at protecting serum potassium.
Okay, let's pull all of this together. If you understand the nephron's baseline physiology, the clinical pharmacology becomes totally intuitive. Loop diuretics trigger massive diuresis by blocking the loop. Thiazides offer moderate diuresis for hypertension in the distal tubule. And those downstream potassium-sparing agents? They provide a weak diuretic effect, but act as crucial physiological shields for your patient's serum potassium. You'll definitely want to take a mental screenshot of this summary right here.
And finally, Section 5, High-Yield Exam Review, 5 Must-Know Concepts.
All right, here are your top 5 high-yield revision points for the boards. Number 1, principal cells reabsorb sodium and water while secreting potassium, and that's driven entirely by the Na+, K+ ATase pump. Number 2, aldosterone is the master regulator. It stimulates P cells to ramp up sodium channels and pumps, and it stimulates alpha intercalated cells to secrete acid via the H+ ATase pump. Number 3, loop diuretics, like furosemide, block the Na2-CLK cotransporter for explosive diuresis, again, perfect for pulmonary edema. Number 4, thiazides inhibit the NaCl cotransport in the distal tubule and are your go-to first line for hypertension. And number 5, K+ sparing diuretics target those P cells in the cortical collecting duct, giving you weak diuresis, but crucially, saving your patient's potassium.
Before we wrap up today, I want to leave you with a little provocative thought experiment to test your newly integrated understanding of the nephron. What if a patient is prescribed both a powerful loop diuretic, which dumps sodium downstream and loses potassium, alongside a K+ sparing diuretic? What exactly happens to their net potassium balance at the cellular level? I want you to really think through those transporter mechanisms we just covered and trace the flow. Thanks so much for diving deep into renal physiology today, and thanks for learning with the explainer. Catch you on the next one.
Welcome back, second-year med students. Today, we're diving into a really fascinating and, notoriously complex area of renal physiology. In this explainer, we're going to look closely at the precise mechanisms the kidney uses to concentrate or dilute urine. We're treating this like a logical, step-by-step masterclass, breaking down the exact physiological machinery that makes this vital process entirely possible. Here's our roadmap for today. We're covering 1, excreting concentrated urine, 2, the countercurrent system setup, 3, building the osmotic gradient, 4, the countercurrent multiplier, 5, ADH and tubular permeability, and finally, 6, the urea recycling mechanism. Let's start with Section 1, excreting concentrated urine, the mammalian survival mechanism. The ability of the kidneys to excrete urine that's more concentrated than plasma is absolutely essential for the survival of mammals living on land. Think about that for a second. As terrestrial beings, our physiological default is constant water loss. This singular ability to hold on to water by concentrating waste into a smaller fluid volume, it is literally what keeps us alive in a dry environment. So, how is this incredible feat functionally achieved? Well, it all comes down to one primary physiological requirement: the maintenance of a highly specific gradient. Now, this isn't just any gradient. It features increasing osmolarity, and it is precisely located along the medullary pyramids of the kidney. Without this escalating concentration of solutes driving deep into the medulla, concentrating urine would be physically impossible. No gradient, no water retention. Moving to Section 2, the countercurrent system setup, multipliers and exchangers. Now, there is a strict functional dichotomy here between producing and maintaining this gradient. This is a classic high-yield concept you absolutely must know for the boards. The loops of Henle, they act as the countercurrent multipliers. Their active operation is what actually produces the gradient. On the flip side, the vasa recta act as countercurrent exchangers. Their job isn't to make the gradient. Instead, their slow blood flow is perfectly designed to maintain it, making sure the blood supply doesn't just wash all those hard-earned solutes away. For this beautiful system of multipliers and exchangers to actually work, it's got to follow strict physical laws. First, the inflow must run parallel to the outflow. Second, it has to be in very close anatomical proximity. And third, the inflow runs in the opposite direction to the outflow, which of course is where we get the term countercurrent. Keep in mind that this precise anti-parallel flow applies to both the loop of Henle and the vasa recta. The physics of it simply won't work without this exact anatomical setup. Section 3, building the osmotic gradient, depth and concentration. Let's isolate the exact directionality and composition of this gradient. Location-wise, it extends all the way from the corticomedullary junction down to the very tips of the medullary pyramids. But what's it actually made of? Well, it's driven by 3 primary solutes: sodium ions, chloride ions, and urea. These are your crucial building blocks. They create the osmotic pressure that we're going to need to pull water out of the collecting ducts a bit later on. We can actually see how this builds by looking at the rule of medullary depth. It's a really elegant, direct anatomical to functional relationship. Step 1, the longer the overall medullary depth. Step 2, the
the greater the length of the loops of Henle. Because these loops are physically longer, step 3, a much higher osmotic pressure gradient can be reached all the way at the tip of the pyramid. And therefore, step 4, you have vastly more chances to excrete a highly concentrated urine. Basically, deeper pyramids equal better water retention.
This brings us to Section 4, the countercurrent multiplier, active transport mechanisms. All right, let's look at the critical molecular machinery. The operation of each loop of Henle as a countercurrent multiplier depends entirely on active transport. Specifically, we're talking about the sodium-potassium 2-chloride or NKCC2 exchangers. Located exclusively in the thick ascending limb, these transporters are the heavy lifters, responsible for actively pumping sodium and chloride right out of the tubule. And what's the direct consequence of all that active transport? It creates a massive osmotic pressure gradient between the fluid left inside the ascending limb and the surrounding medullary interstitium. By forcefully evicting those ions while remaining completely impermeable to water, the thick ascending limb is literally splitting the osmolarity. It's loading up the interstitial tissue with the exact solutes needed to draw water out later. Let's move on to Section 5. ADH and tubular permeability hormonal logic. Now, having a highly concentrated, salty medulla just isn't enough on its own. A high level of ADH, or antidiuretic hormone, is the next basic requirement for forming concentrated urine. Without high ADH, that beautiful osmotic gradient we just spent all this time building in the medullary interstitium, it's essentially useless because the final segments of the nephron simply won't physically interact with it. This brings us to another classic high-yield physiological concept, the differential effect of ADH. In the cortical and outer medullary collecting ducts, ADH acts to significantly increase water permeability by inserting aquaporin channels. But, and note this carefully, ADH does not increase urea permeability in these specific outer segments. Water can leave, but the urea cannot. So the really crucial point here is tracing this sequential physical logic. Because of that differential permeability, step 1, water is rapidly reabsorbed from the most cortical and outer medullary collecting ducts, drawn right out by the interstitial gradient. Step 2, the urea cannot follow. It remains trapped behind in the tubular fluid. Therefore, step 3, as the fluid volume drops but the absolute amount of urea stays exactly the same, the urea concentration inside the tubular fluid dramatically spikes.
And finally, Section 6, the urea recycling mechanism, the second contributor.
Remember a bit earlier when we said the gradient was made of sodium, chloride, and urea? Well, here is where the urea finally joins the party. Urea recycling from the inner medullary collecting ducts is the second major process that contributes to establishing that massive cortical papillary osmotic gradient. The kidney doesn't just excrete all that trapped, highly concentrated urea. It purposefully leaks some of it back into the deep medulla via facilitated diffusion through specific urea transporters to reinforce the hyperosmolarity.
Which leaves us with a compelling question to wrap up part 1 of our analysis. If ADH increases water permeability in the outer ducts, but purposely leaves urea trapped to concentrate it, what critical shift happens when this supersaturated fluid finally reaches the inner medullary collecting ducts, where the effects of ADH inherently change? Think carefully about how that permeability might shift to allow that urea to actually be recycled. Keep that physiological cliffhanger in mind as you review your notes on tubular segments. Thanks so much for joining me for this explainer, and I'll catch you next time.
Welcome to the Explainer. Today we're taking a high-yield, deep dive right into the complex mechanics of renal physiology. If you're a second-year med student trying to lock down exactly how the kidney handles ions, regulates fluids, and responds to some pretty intense hormonal feedback loops, you are absolutely in the right place. We're going to connect the dots all the way from cellular transport up to systemic blood pressure. Let's get started. Here is our roadmap for today. We'll be hitting sodium and urea excretion, potassium and acid-base handling, ECF volume regulation, forces modulating tubular reabsorption, hormonal control, and we'll wrap the whole thing up with a high-yield exam review.
All right, let's jump straight into Section 1, sodium and urea excretion.
So our understanding of macro-level extracellular fluid volume really begins with how individual nephrons handle key solutes. Before we get into the heavy lifter, which is obviously sodium, let's establish the baseline for urea excretion. It's actually fundamentally determined by just 2 main factors: the concentration of urea in your plasma and your glomerular filtration rate, or GFR. Simply put, more urea in the blood and a faster filtration rate is going to equal a much higher rate of urea excretion. Pretty straightforward, right? Okay, let's dive into exactly where all that sodium is going. It's absolutely crucial to know these numbers. In the proximal convoluted tubule, a massive 65% of filtered sodium is reabsorbed right off the bat. Moving down the loop of Henle into the thick ascending limb, we get another 27% reabsorbed. Now, by the time we hit the distal convoluted tubule and the collecting duct, we're dealing with single digits, 4%, 3%, and finally less than 1% actually excreted. But don't let those small distal numbers fool you. That is precisely where the final, highly regulated fine-tuning of your body's sodium balance takes place.
To see that fine-tuning in action, let's focus in on the principal cell, or P cell, in the distal nephron. Picture aldosterone entering the cell from the blood. It binds to its receptor and initiates transcription right inside the nucleus. This mRNA translation leads to the synthesis of brand new proteins. Specifically, we're talking about new sodium and potassium channels popping up on the apical or luminal membrane and new ATP pumps being built on the basolateral membrane. This beautifully coordinated synthesis ramps up sodium reabsorption into the blood and, at the exact same time, pushes potassium out into the lumen to be secreted.
Moving right along, we have Section 2, Potassium and Acid-Base Handling.
Now, what's really interesting here is the delicate interplay of gradients driving this potassium secretion. Potassium is actively pulled into the cell from the interstitial fluid by that basolateral NAK-ATPase pump. It then exits into the tubular fluid through those apical potassium channels. This movement is driven by a strong chemical gradient, because intracellular potassium is high, but it's also heavily dependent on the electrical gradient. You see, sodium influx through its own apical channel makes the lumen more negative, and that electronegativity acts like a magnet, actively pulling the positively charged potassium right out of the cell.
So what does this mean clinically? Well, because of these specific transport mechanics, several distinct conditions will predictably increase urinary potassium secretion. A high potassium diet or a high plasma concentration will naturally boost secretion, but check out the others: aldosterone excess, alkalosis, the use of diuretics, high tubular sodium, or excess tubular anions. The absolute key takeaway for your board exams is that an increase in sodium delivery to the distal tubule, like when you give a patient loop diuretics, will accelerate that apical sodium-potassium exchange, leading to some very significant potassium loss. Shifting gears to acid-base handling, let's carefully trace the 8-step pathway for hydrogen ion secretion in the proximal tubule. Step 1, we've got the vital NaH antiport secreting hydrogen into the lumen. In Step 2, that hydrogen combines with filtered bicarbonate to form CO2 and water. That CO2 rapidly diffuses into the cell. That's Step 3, where carbonic anhydrase gets to work reforming hydrogen and bicarbonate. The new hydrogen gets recycled right back into the lumen, while the new bicarbonate is reabsorbed back into the blood at Step 5. Furthermore, if you look at Step 6, glutamine is metabolized into ammonium and even more bicarbonate. This provides an incredibly powerful alternative route to excrete acid and buffer the blood. All right, zooming out a bit, here is Section 3, Regulating Extracellular Fluid Volume. Let's look at the macro-level homeostatic reflexes. We can break this down using some simple if-then logic. If blood pressure and volume decrease, then your volume and baroreceptors trigger a dual response. The cardiovascular system increases cardiac output and vasoconstricts to raise pressure, while behavioral centers trigger thirst, and the kidneys actively conserve water. Conversely, if blood pressure is elevated, then the cardiovascular system vasodilates, and the kidneys actively excrete salts and water to reduce that extracellular fluid volume and bring your blood pressure safely back down. And when that blood volume is elevated, it literally physically stretches the atrial myocardial cells, which triggers this beautiful endocrine cascade. The stretched cells release natriuretic peptides. Think of these peptides as a systemic override switch hitting multiple organs simultaneously. They hit the hypothalamus to decrease ADH, they target the kidney to increase GFR and drop renin, they hit the adrenal cortex to decrease aldosterone, and the medulla oblongata to decrease blood pressure. The whole shebang converges on one massive goal: increasing sodium chloride and water excretion to rapidly relieve that volume overload. Which brings us to Section 4, Forces Modulating Tubular Reabsorption. So we've got two baseline physical forces to consider first. Number 1 is osmotic diuresis. This is when unabsorbed solutes, like glucose in an uncontrolled diabetic, stay in the tubule. They osmotically hold on to water, driving up urine output. Number 2 is plasma colloid osmotic pressure. Higher protein concentrations in the peritubular capillaries will physically pull more fluid back out of the tubules and into the blood, increasing your overall reabsorption.
But here is the absolute crucial point. How does the nervous system override those physical factors? Well, sympathetic stimulation via beta-1 receptors acts through two very distinct steps. First, it directly constricts the afferent arterioles. This decreases glomerular pressure, drops the GFR, and thereby decreases urine output. Second, those exact same beta-1 receptors stimulate the juxtaglomerular complex to release renin. That renin forms angiotensin II, which further decreases urine output. Both of these pathways effectively converge to powerfully retain fluid during a fight-or-flight stress response.
So what happens if arterial pressure just spikes, assuming autoregulation is absent or failing due to disease? Well, an increase in arterial pressure actually works from two different angles. First, it increases glomerular pressure, which directly increases GFR and therefore increases urine output. Simultaneously, it increases the pressure within the peritubular capillaries. This physically opposes tubular reabsorption, causing even more fluid to stay right there in the tubule. Both of these physical pressure pathways ensure that high arterial pressure forces the kidney to ramp up urine output.
Let's tackle Section 5, hormonal control of reabsorption. This is heavily tested, so pay close attention.
Let's quickly summarize the direct actions of our first two key hormones here. ADH, whether we're talking acute or chronic effects, inserts water channels into the collecting ducts to strongly decrease urine output. Then we have aldosterone. Remember that principal cell we looked at earlier? Aldosterone explicitly increases sodium reabsorption while actively increasing potassium and hydrogen secretion in the distal and collecting tubules. By pulling sodium back into the blood, water naturally follows and your urine output drops. And then, of course, there's angiotensin II. This thing is a literal multi-site powerhouse. It does not just do one thing. It stimulates aldosterone secretion. It directly stimulates both the NAK-ATPase pump and the NHE exchanger in the proximal tubule. It powerfully constricts efferent arterioles, which preserves filtration while dropping peritubular pressure to maximize reabsorption. Oh, and it constricts mesangial to reduce the capillary filtration coefficient. It completely synergizes its actions across all these cellular targets to dramatically decrease urine output and boost your fluid retention.
Finally, Section 6, high-yield exam review. Let's bring it all together.
Let's lock down the absolute must-knows for your exams. Ready? 1. Urea excretion relies heavily on GFR and plasma concentration. 2. P cells in the distal nephron are your key mediators for aldosterone-driven sodium reabsorption and potassium secretion. 3. Proximal hydrogen secretion relies fundamentally on the NAH antiport and glutamine metabolism. 4. Sympathetic beta-1 receptors forcefully decrease GFR and trigger the release of renin. And 5. Angiotensin II is a systemic heavyweight operating at multiple nephron sites simultaneously to retain fluid.
Before we wrap up, I want to leave you with a massive practical tip. Always distinguish between apical transporters, the ones facing the lumen, and basolateral pumps, facing the blood. When you're analyzing a hormone like aldosterone or angiotensin II, knowing exactly which side of a cell it acts on changes everything. Just think about it for a second. How could a defect in just one of these localized microscopic cellular pumps entirely unravel a patient's systemic blood pressure loop? Keep that provocative question in the back of your mind as you review your textbook. Thank you so much for joining me on this explainer, and happy studying.